Hi, welcome to Anatomy Lab of the Cell. And so basically in this section, we're just going to use two different cell models and just go through the various structures as you learn about in lecture. So as you know, the outer part of the cell is known as the plasma membrane. So it's just going to be the outermost part on a model. And realize that you have pictures of both models here. So even though I may be pointing to almost all of these structures on one model, you then need to look at the picture of the other model and find all the structures on the other model. And also go through your list of terms. You will notice that all of these are just following your list of terms. Okay. So inside the center of the cell, we have the nucleus represented by this big area and the bracket. The nucleus has an outer envelope. You can call it the nuclear membrane or the nuclear envelope. And in this, there are openings known as nuclear pores. Also, in the center of the nucleus is this orange structure, is a nucleolus. Now, I want to point out the spelling of nucleus and nucleolus, and that there's only 2 letters difference between the two. So you need to write the correct spelling for nucleus and the correct spelling for nucleolus in order to get any credit for these 2 terms. Even on a quiz and a test, I'm not going to let you have the 1 extra letter, the L, for a nucleus with an extra L in it. Only nucleolus has 2 L's. Now, if you recall from your lectures, what is attached to the nucleus is going to be organelles floating in the cytosol. So any of this whitish background material is going to be cytosol. And the first organelle is the rough endoplasmic reticulum. You also can see it right here adjacent to the nucleus. How can you tell it's rough endoplasmic reticulum? Because you can see the little attachments, the little studs on the external surface. In comparison to this part, which is smooth, so this without the bluish stuff on the outside with the little studs, that would be smooth endoplasmic reticulum. So those little studs are the ribosomes. So ribosomes can be pointed to when they are attached to the rough ER, or they can be pointed to when they are just floating in the cytosol, and either one of those would be identified as a ribosome. The next is the Golgi apparatus. If you recall, it's like a bunch of flattened pancakes, stacks put together, so that's the Golgi. And then pinched off the Golgi are these little vesicles, which are going to carry substances throughout the cell. This orange structure, whether it is looking at the external surface like this, or whether it's like these, which are cut in half, each of these is a mitochondrion, because you clearly can see the cristae of the mitochondria inside of them. Now, this next structure is a lysosome. And before students say, well, the yellow one's a lysosome and not the gray one, or the doesn't really matter. The only other thing we're learning about is the lysosome. We're not learning about peroxisomes, anything else, so it doesn't matter which one I point to. Your only other option is the lysosome. And then if you recall, the 2 sets of microtubules that are perpendicular to each other, each of these is a centriole. Looking at the other cell model, so go through on this cell model and try to find all the structures we just covered on the other one. This is a cell model which shows the little surface projections, each of which is a microvillus, and notice there is no cytoskeleton inside them. But if you look here closely at the side of this model, what you clearly can see are some little cytoskeleton things. So these are showing the location of desmosomes. And with that, we are done with the cell for lab. Thank you, and I'll see you again soon for the next video.